We go.
Yeah.
Good afternoon, good morning, good evening, wherever you are. Welcome to ECTRIMS 2015 in Barcelona. So just to introduce myself, I'm David Baker. I'm from Barcelona, London in the UK, and we're here today to talk to you about what's been going on and what's exciting, what's hot and what's new in ECTRIMS 2015. And on my right, we have Shamali. So please introduce yourself, Shamali.
So my name is Shamali. I'm one of the consultant neurologists at Barts Health NHS Trust. I'm also an avid blogger. So if you follow our Barts MS Research blog, my mnemonic is NDG, and I sometimes stick to the periphery, but sometimes I can be quite controversial on what I think about new treatments coming into MS. So that's one of the things which I do like to discuss today and round up on what I think are clinically important for MS over the next year. So the way we're going to run the blog is we will start off going through sections of the ECTRIMS roundup, and also we'll be answering questions as they come up on our Twitter sites, on our web pages, as well as any questions or queries from our audiences.
So first of all, if you want to have a question, you can do it obviously by joining the Hangout, but also you can send us a tweet. And the tweet is at Barts MS blog. So if you do that, I'll be able to see it, and hopefully we will be able to answer your questions. I guess you also have to appreciate that there's been quite a lot of things going on in the last few days, and we haven't seen everything, and I guess nobody can. At the moment, there's a whole host of people missing this excellent event, but nevertheless, you will be able to watch it on YouTube, so you won't miss out on all of the fun. So let's start the show then, shall we? And we also have an audience, and if they think we're talking rubbish or anything, they can interject, and hopefully maybe we can even get them on stage to also introduce themselves. So, okay, Shamli.
So let's start with the plenary session. It was done by Professor Alan Thompson from UCL Queen Square Institute of Neurology. And Alan put to the forefront progressive MS. So if you're aware of Alan's background, he is very much into neuroprotective therapies and also about targeting the molecular side aspects of progressive MS. So he, in fact, highlighted some of the accomplishments which the society has made in terms of the Progressive Alliance, as well as also his own group. So he highlighted Raj Kapoor's neuroprotective therapy, lamotrigine in secondary progressive MS, which you all know initial findings were negative in terms of the primary brain atrophy outcome, but further subgroup or post hoc analysis has showed that there is actual improvement in walking times in those who were actually on lamotrigine treatment versus placebo. And also many of you will be aware of my work on neurofilaments, which showed that neurofilaments level reduced in lamotrigine treated patients with a treatment lag of 1 year from inducement of the treatment therapy. So that was promising. He also mentioned adaptive trial designs for neuroprotection, in particular the MS SMART design, which uses 3 drugs versus placebo to see if any of these will have neuroprotective effect in secondary progressive MS.
So what are those 3 drugs?
So riluzole, which may have antimicroglial blocking action, and also amiloride, which is a calcium channel blocker. So the lamotrigine drug, which I mentioned previously, is a sodium channel blocker. Amiloride will work around the same concept, and also fluoxetine, although I don't know exactly what fluoxetine accomplishes.
That's Prozac. I didn't know it was called Prozac, but it's Prozac. It's you or me.
And the idea is they will recruit about 600 odd patients on this, and we'll see if any of these treatments actually have a neuroprotective effect. So just to highlight, this is de novo neuroprotection. So all of the patients entering into the MS SMART study are on anti-inflammatory treatment. They also may have immune pathogenesis in their disease group, but these patients aren't on anti-inflammatory treatments. So that was Raj's work. There was also additional work on the phenytoin study in optic neuritis, which was recently highlighted again this morning by Gavin Giovannoni, which is to do neuroprotection very early on. So if you imagine optic neuritis developing, they introduced phenytoin within 4 days of the patient becoming symptomatic and developing optic neuritis.
So that's kind of a theme that was, I guess, introduced at the very beginning of the meeting, the whole concept of brain health and trying to treat early to deal with the problem. So I think obviously the question is ECTRIMS has often been seen as a meeting about relapsing remitting disease. I think this is probably one meeting where there is a change, because we are going to hear some good news, I think. It hasn't been shown yet, but we've saw a press release. So I think the tide is changing and things are starting to work for progressive disease as well. So do you want to say anything about the brain health thing, Shamali? Or
yeah,
not?
so why not? Let's track to the forefront of what we're doing, which is Gavin Giovannoni in particular has highlighted that people sign up to the brain health in MS. So it is neurologists versus MS. That's what we're talking about. And it's very apparent in that session that the treatment has to happen very early on. So do not wait to treat the MS, start very early. It was also interesting from the OPERA results that there was a 4-year lag before treatment, which is quite substantive, and it highlights that in parts of the world with MS, you don't actually have treatments available. So Gavin highlighted that there are generics or biosimilars which are available, which are off patent, which can be used in countries which don't have access to the injectables or to the newer therapies as they come in. So the list is not unfamiliar to neurologists. They are all the immunosuppressants which we use, such as azathioprine, even cladribine, which is being resurrected but can be used off patent to target highly active relapsing remitting MS.
So what you're saying is, if you can't have access to the expensive drugs, then there may be something that your neurologist can do. But one thing that we do need to do is obviously focus on the saving brain. And the best way to do that is to make sure that the brain doesn't get damaged by multiple sclerosis. So what we want to do is to treat a target, and the target is really no evidence of disease activity. And if we can achieve that, then I think the data has been shown. There has been quite a number of posters that have been shown here that you can have quite significant impact. And I don't know if you saw this, Shamali, but there was some data presented with alemtuzumab today looking at 10-year follow-up and even a 20-year follow-up of people who were taking alemtuzumab in the very early trials. Some good news and some bad news. I think what was evident was if people had relapsing remitting disease and they were taking drug quite early, that it looked like their disease was really sort of stabilizing. There were more people doing better than people doing worse. The downside was that people with secondary progression tended to still progress. So again, it's this take-home message that really what we want to be doing is to ensuring that we treat as early as possible to stop secondary progression from occurring. So this is one of the themes within the Time is Brain, and there is a website, and if you go on the MS Society blog, oh, it's in backwards. That's the power of cameras.
Can I just point out where the UK is? It's right at the bottom. Poland is the only other country other than the UK which doesn't prescribe neuroDMTs. Now, I have a question for you as a scientist, David. What do you think patients would like? What do you think patients would like on us neurologists?
Well, I'm the mouse doctor, if you've not heard of me before. But I can tell you what my mice like. My mice like us to get in the disease under control as quick as possible. And if we do that, we stop them developing progressive disease, and we then can actually halt further nerve loss. Because with each attack, there is nerve damage, and I think there's been a number of posters again that clearly show having a lot of relapses early on is not good for you. And so we really need to work out how best we can get that under control. Now, one thing that has really come out at the ECTRIMS meeting is it's going to be much more difficult for you guys as neurologists to decide what to do, because we're getting more and more drugs that actually are showing some efficacy. So there's a new one today, and I think we should probably start off with that. One of the big things of ECTRIMS 2015.
Before we discuss the OPERA studies, can I find out in the audience here, how many of you treat at the CIS stage? How many of you? So we have one show of hands, or second show of hands, third. Okay, we're growing in number. And
CIS, clinically isolated syndrome, for you at home, in case you didn't know what it was.
Okay. And how many of you wait for established MS to develop before you treat?
Too embarrassed to put your hands up. Nope.
How many of you, given the opportunity, would like to treat MS at the onset of disease?
A lot.
Okay, so these are important things. I know we don't really, we skirt around the radiologically isolated syndromes, and it probably needs to be discussed at some stage, but I don't think as neurologists we're there yet to discuss how to treat radiologically isolated syndromes. But if you're following the NEDA concept, that's no evidence of disease activity, you should be thinking about treating radiologically isolated syndromes at a future stage. The fact about radiologically isolated syndromes may be made much easier if they're oligoclonal band positive. So if they have OCB positive MS or IgM bands, then they're more likely to convert to true MS and more likely to develop a disabled disease. So that may make it easier. So the development of stratification using prognostic biomarkers may help with the management of MS much earlier on.
It also helps if you live in a country where neurologists are able to treat at the early stages of disease. I think in the UK, we're lagging behind many other countries. So, you know, that's another thing, I guess. There is a regional variation. Some people, places clearly do. And there were some posters, again, I saw one with our badger, again, treating at CIS. And, you know, it slowed down the conversion rate to clinically definite MS. So if we can treat at these early stages, I think, you know, again, progress can be made, and there's more data showing that type of thing, and we've seen it this week, and we'll probably see it more.
We've had a question on our tweet, or was it on our blog, about what about secondary progressive MS? So let me just say that being clinicians and being scientists who blog regularly, this is a very recurring, highly recurring theme from our viewers, from the people who follow our blogs. So just to backtrack, so the secondary progressive MS and progressive MS is becoming the focus of management. And I believe with the next ECTRIMS being in London, this is going to probably most likely form the main component of the meeting. So neuroprotectants, there are several ways of doing neuroprotection. There is one way, which is to go de novo with a neuroprotectant at either early stage of MS or in the early stage of secondary progressive MS. Currently, the only treatments which you may consider for later stages of secondary progressive MS would be reparative therapies. And at the moment, we may have anti-lingo, which may work, but it's only been trialed as a focal model of remyelination. So if you look at earlier phases of MS, you need to be thinking of channel blockade for neuroprotection and reducing the nitric oxide production, as well as the calcium channel or calcium overload, which you see within axons. Now, there are two ways of doing neuroprotection in the early phase of MS. There is that de novo way, which is where you go with your neuroprotectant, or you take out the inflammatory phase of MS and piggyback on the actual neuroprotectant. Which strategy will work is not known. So there are two competing clinical trials at the moment, which is the MS SMART study, which is treatment naive or treatment no inflammatory agents are actually introduced, which is the MS SMART. And then the Proxima study, which has patients previously on anti-inflammatory treatments, and also then on top of it, they receive a neuroprotectant. So those are the two trial designs which you could expect to use when you use neuroprotection. So that hopefully will do a readout in the next 2 years in terms of trial outcomes.
Yeah, so I think the take-home message is certainly for people with progressive MS, you're not forgotten. In fact, you're really at the forefront of really the thought process. But the downside is, you know, it will take time for those treatments to filter through. So we have to wait till they do report, you know, and obviously then it takes time to deliver the drugs to you. So I think we should go on to a drug that should be able to be delivered to you reasonably soon, and that's the results of the OPERA. And what do I mean by OPERA? It's just the name of a trial with a new drug, which you'll get to hear about in the coming future, and this is called Ocrelizumab. Now, Ocrelizumab is a monoclonal antibody that targets B cells. So for many years, you know, we've had the people working with the animal models telling you, you know, MS is a T-cell disease. However, we've seen the results today of an antibody that blocks B cells, and it's pretty impressive, I think. And I think from looking at the data, we can see it is as potent as the current potent drugs. So it's a new, very effective treatment. The beauty of the treatment compared to some of the other drugs is that there aren't some of the side effects as or haven't been seen yet. Obviously, it will depend with time as people take the drug for longer. However, we could say, I think, that the drug Ocrelizumab was probably as effective as alemtuzumab in terms of blocking the relapse rate. It was about 50% down compared to people taking beta interferon, and really what I noticed from the graphs is, you know, and some of those failures were probably early on, and so, you know, it probably takes a few months for the drug effect to really come through, and so some of those failures were seen within the first, you know, 12 weeks or so, and maybe the results would be more impressive if you kind of thought about what is the effect of being as a re-baseline. However, it was effective, and against gadolinium enhancing lesions, yes, it was very remarkably effective.
So it established about a 94% reduction in gadolinium enhancing lesions, as well as 72% reduction in T2 volume lesions early on, but at later stage it also went down to almost full complete block in T2 lesions. The study wasn't powered for clinical disability endpoints, but did manage to meet it with a 40% reduction in the chances of you becoming disabled versus being on interferon beta. So like alemtuzumab, they say that this targets the CD20 in the intermediate phase of T-cell differentiation. So it doesn't affect the stem cell pool of the B cells, neither does it affect the plasma cell pool of the B cells. So you may, in fact, only be affecting the circulating B cells, which is about 2% of B cells, with the drug ocrelizumab. Now, what I'd like to ask David is, why don't we just use rituximab?
Whoa, now that's an interesting one. Rituximab is also an anti-CD20 antibody, and it has been used in MS. And in fact, there's a lot of posters, again, at ECTRIMS, looking at over 1,000 people. And it does show that it can inhibit relapses. So I think as a class of drugs, I think there's no question that the antibodies that deplete these B cells can have an effect. Now, what's the difference between the two? Well, rituximab, as the name tells you, actually is what you call a chimeric antibody, and it means it's half mouse, or part of it is mouse, and the other part is human, which means when you inject protein over and over again, the mouse bits can be recognized as being foreign, and so eventually you can get antibody responses against them, which can cause injection site reactions or allergic shocks, perhaps. So what ocrelizumab has done is it tries to avoid those processes by genetic engineering. So there's only a tiny bit of the mouse antibody, and so it should have a better side effect potential, because it obviously shouldn't cause adverse injection site reactions. So that's one difference, and maybe subtle differences in also which in epitope of the CD20 it binds to.
So there was a mention, and it's a recurring theme. They said that it's not working by cytokine mediated toxicity. It's going to be more suppression of the cell mediated version. So that's supposedly the difference you get from changing the epitope. But we must realize that rituximab is more freely available. The question is whether ocrelizumab may actually share the PML risk of rituximab, which at the moment is difficult to say, but we'll know soon enough.
I mean, I think obviously PML has been noted with rituximab. It hasn't been seen in the ocrelizumab. In terms of the side effects, I think the side effect profile was reasonably good. It didn't show the autoimmunities that are very frequent with alemtuzumab. The main type of side effects were probably a few infections, which would be kind of anticipated with this type of drug. But I think compared to some of the other drugs out there, the side effect profile is better, and the level of efficacy is just as good. So it's going to be interesting to see what happens. But obviously, it has to be approved. And so there were 2 trials done, and I'm sure they'll be going to the regulators, I think, in 2016, and it may get approved. And once it does get approved, then it will be, doctors won't be able to really prescribe rituximab anymore, because there is a licensed alternative. And once there is a licensed drug out there, that's what the neurologists will be obliged to use, because otherwise they're at risk from things such as their insurance. They may not be covered, and also the payers may not pay for the drug if it's being used off-label when there is a licensed drug available. So, you know, in time, maybe it would be difficult to use rituximab.
So one of the questions already asked is, so the results look similar to alemtuzumab. Why don't we just simply use alemtuzumab? Now, alemtuzumab has its 5-year data. So if I put the question to the audience, who would pick ocrelizumab if they had the option of prescribing ocrelizumab in relapsing remitting MS? By a show of hands. And who would pick alemtuzumab?
So ocrelizumab wins.
By one, dare I say. Alemtuzumab, what is the difference between alemtuzumab and ocrelizumab? Well, alemtuzumab targets T-cell population. So maybe the best effort might be to smorgasbord bomb the immune system with both drugs. But in fact, I think the toxicity of the combination treatment might be phenomenal. Alemtuzumab is also only 2 courses. So whereas this you'd have to renew on a regular basis by the looks of things. But looking at the level of suppression of gadolinium-enhancing lesions, you may be able to push the intervals of injections much further than what has been done in the trial. So how does alemtuzumab work?
Well, alemtuzumab obviously depletes T cells, and it depletes B cells too. So it has, a similar mechanism in that sense. I mean, the main difference, of course, is alemtuzumab is available now, and ocrelizumab isn't. And we had an example of a person who was, potentially in line for a treatment and didn't get the treatment, and it was delayed for 2 months, and this person had a devastating relapse, and they were left with a problem. So, you have to think about, if you're in that position, what you do here and now, rather than wait for around the corner, because the grass will always be greener around the corner. I can assure you that. So we have to deal with the tools that we have available now, I think.
I do think we need to wait for the cell immunology studies on ocrelizumab. They'd be interesting to see. Alemtuzumab brings T cells to a nadir within definitely by day 7 of infusion, almost by 98%. They start to repopulate within a month. The degree of repopulation is very much dependent on CD8 repopulating much quicker than CD4. There seems to be a delay in CD4 repopulation, and the B cells also repopulate, but ahead of the regulatory T cell pool. And that's where you see all of the systemic autoimmune disorders coming through. Ocrelizumab is targeting now B cells predominantly. So you're looking at B cell antibody-mediated response. You're also looking at the B cell follicles, which are there triggering the microglia. So that's another target they'll be working in. And also the main role, which is their antigen presentation to T cells, so the maintenance of the autoimmune process. So the two very different drugs. The way you choose to put which patient on which may well depend on what their phenotype of disease is. So that's definitely an area of work which needs to be explored to see what the dominant pattern of immunopathogenesis in the patient's MS is.
Yeah, I mean, this drug is very interesting. It opens up the big kettle of fish, really. What drives multiple sclerosis? Is it T cells? Is it B cells? Is it the virus in the B cells? And, it's been suggested that maybe that's what this drug is doing. It's getting rid of what drives the disease. Time will tell. You mentioned alemtuzumab, and what we've heard is the 5-year data. I don't know if you saw the 5-year data.
Yes, so the 5-year data of the extension study of the CARE-MS-1 shows that it does what we expect it to do. So majority of disease activity was suppressed at year 5, as it was on the 4-year data, and the brain volume loss or brain atrophy rate was also maintained at year 5, slightly better than it was at year 4, but maintained. Now, what you have to realize is that the brain atrophy rate is now back to normal population brain atrophy rates. So a normal person atrophies at about 0.3 to 0.5% per year, whereas an MS brain can lose brain volume at about 0.5 to 1%, in some cases much greater than that. And what this does is it normalizes back to normal background levels. And this has implications, obviously, for achieving disability outcomes much later on in the disease course than immediate outcome benefits. So that needs to be considered. I don't think there is a comparable drug at the moment in the market which can achieve this as effectively as alemtuzumab is demonstrating for an induction treatment. The future of MS treatments is going to be induction treatments. We can't have, in this day and age, have patients coming back on a regular basis where our monitoring requirements are increasing rapidly, where we're also seeing them again back and forth for reviews of their medications. The other thing is the Ocrelizumab data, which we should be hearing on Saturday morning as breaking news for the primary progressive patients, which has also met its disability outcome. But again, that's preliminary data, and you have to realize that's time to disability in 3 months.
But the good news is, obviously, it's the first drug that's really shown to have a significant impact on primary progressive MS. So again, I think this is where the game starts to change. The big question we all want asking, and we don't know the answer, is, does it affect disease in everybody with primary progressive MS? Or is it only really targeting the people with gadolinium enhancing relapsing primary progressive MS? And I suspect, well, I know what the answer will be, but we have to kind of wait and see until the data is presented, which is tomorrow morning. So unfortunately, we can't really talk about that. But we do know from the press releases that there is about a 25% reduction in the rate of progression, or 20% or something like that. And so it's a start. And I think we think about biology, we can think, well, we do want to stop inflammatory attacks, but probably we have to layer things on top. This is where the neuroprotection and the neurorepair will come in. And the good news is it's a positive result, and we can work from that. You know, for many years we had the beta interferons, which were only, you know, about 25 to 30% effective, and now we're probably looking at sort of 70, 80% effective. So you know, the ball has started to change, and I think we'll be seeing more and more of these, because it tells us that we know how to do trials in primary progressive MS. And it's not that we haven't got ideas about what drugs to use, it's just how best to show them working.
Now, I'd like to move on away from the clinical trial data to some of the other parallel sessions. One of the things which I found interesting, and probably we'll hear a lot more about, is the gut microbiota. How many of you actually attended that session? Or, yeah, so you know what I'll be talking about. Now, this is interesting because we often ignore what's going on in our guts, but there were some ugly looking pictures of how to recycle your own microbiota, and that's something which I get asked about, fecal transplants and all of these things. So the concept of the gut microbiota is that all of us by puberty have a stable microbiome system in our guts, and what this microbial phenotype does is it kind of triggers or decides what our MS immune system might be like. So if the immune system is active, it would actually go and trigger systemic autoimmune diseases. So they're hypothesizing that this may be reflective of how MS may develop. And there are some controversial data about this. Now, there's some interesting data on the T-cell transgenic receptor.
Yeah, so obviously it's a bit science, and I guess it could be science fiction as well as science fact, but there were some studies where they have made mice that have got transgenic T-cell receptors, which means that all their T-cells are programmed to target a certain myelin protein, and if you kind of then tickle the cells, they will migrate around the body, and if they get into the brain, then they can trigger an MS-like disease. So I guess what we heard this morning, and Shamali's going to tell us about, is can you change those bacteria in the gut to have an effect in these mice? And what you can do is you can not only use mice, you can clean mice, get rid of their bacteria, and then you can give them human poo. And what happens then?
So this was a twin study which was presented, and effectively they took twins with where one of the twins developed MS and one didn't, and then did a fecal transplant for the mice. And what they found is that these mice who received the fecal transplant from the MS affected twin were more likely to have a more active disease course versus the twin who did not go on to develop MS. So these are now functional studies looking at what actually happens when the gut microbe is different from one individual to the next. Now, the question here is that everyone's going to have a different microbiome, but this may be a large untapped field of MS research where we may be able to find new targets and new therapeutics to modify the disease course in MS. If you consider that bulk, almost 60% of MS is due to environmental factors, then this is a region which we really do need to look further into.
So you're saying we are what we eat. Now, what I would say, there are ways of developing treatments from this, but one of them would be a fecal transplant, and there are people thinking about doing that. But what we would say is there is no evidence that it is useful yet. So do not be paying for treatments where there's no scientific evidence to support that it's actually a valid approach. So I'm sure it will be looked at scientifically, and if it's shown a value, then we can support it. Obviously, until that is the case, we just say it's a bit of ongoing science and watch this space. So, are there any other things that you would like to talk about?
So I'd like to round up our session with a discussion of what I thought were interesting posters. One of the ones, which was from Mark Friedman's group in Ottawa, was, is it possible to predict benign MS? How many of you in the audience actually believe there's such a thing as benign MS? So no one thinks that there is benign MS, but there are attempts to categorize benign MS, and that is using EDSS scores. So if you're less than 2 for at least 20 years, you are considered to have benign MS. But what Mark Friedman's results showed was that there was no clinical variable which can predict whether an MS disease course will be benign or not. What it did say was that if you acquired early disability, that is, you had an EDSS score greater than 2 before 10 years, then you were unlikely to have benign MS. And that would be almost 80% of MS patients. So this is where we're probably not treating as well as we should be in terms of how many patients we start on DMT. The other things is there was an interesting poster from the Swiss multiple sclerosis cohort study, which looked at about 1,000 Swiss patients, where EDSS was increased in all parts of disease: relapsing limiting MS, secondary progressive MS, and primary progressive MS, and that was based on whether they had relapses or not. So their ability to progress in disability in any form of MS was based on relapses. They also looked at to see what the introduction of the newer therapies has done in terms of reducing annualized relapse rates. And they found that it was in the order of natalizumab being highly efficacious, followed by fingolimod, and then much further on the first-line DMTs. So you do actually see that in real-term practice, that the highly active treatments probably have the best effect.
I'd love a cup of tea as well, if I could see you there. Have you got a question? Smile. You're on telly.
Hi, are you talking to me over here in Ohio?
Hi, I'm talking to you. Yeah.
Hey, good morning. It's Laura. I'm sorry, I'm late checking in. My power company quite inconveniently sent a message about a half hour ago. We're going to turn your power off. So my internet has been down. I was looking forward to coming to this Google Hangout, particularly because of the great work that you do. But I want to talk a little bit more about the Ocrelizumab studies. First of all, can you tell me what oratorio and opera really stand for as an acronym?
Oh, God.
I can't find it anywhere.
Oh, God. You know, the way they come out with these names, it's got...
It has nothing to do with singing ability.
No, it's got nothing to do with singing ability. You go through a title like phase 2, blah, 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 blah. You pick out O, and then you look at, there'll be another word with P in it, and then, I don't know. It's something that the companies like to hang on, so we can talk about this trial and we know what we're talking about. I'd have to look on I'll post it on the Bart's MS blog, what they mean.
Okay, yeah, because as I've been trying to blog about this, I'd like to tell my readers what it is that I'm actually talking about, because I know it means something more than getting together and singing for the choir. We do that enough as it is. Can you talk a little bit though about the window of opportunity for treatment that these studies show? Is it a narrowly defined window where the people with RRMS or even PPMS, tomorrow's news, we only have a certain opportunity, a certain space in that little grid where people really might respond? Or is it too early to say that we don't know that?
So we don't know the breakdown of the oratorio, which is a PPMS group, but we do know that from the relapsing remitting opera 1 and 2, the mean EDSS is 2. So we're looking at very early disease course. This is most likely, if you look at epidemiological studies, this is first 5 years from diagnosis. So very early MS. And that would be considered what should be the window of opportunity.
Yeah, I mean, I think, we'll know with data, the best way is when we have these very highly effective drugs and then looking with time, hopefully if we've got a database with that information. I mean, there was a study from the MS base looking about when they believe secondary progression occurs versus relapsing remitting disease, and it was interesting that they suggested it may occur quite a few years before neurologists thought it occurred. But that's going to be, will stand the test of time. Now, unfortunately, I believe we've got to go because our time is up and we're going to get kicked out of the room.
So it's not tea. It's not tea because I'm in the U.S., but have a great afternoon. Enjoy Barcelona and have a glass of sangria for us, please. And thanks for all you do.
Normally, I'll be off for a glass of sangria. So, or 3. So, for me anyway. So, it's up to me to thank you for tuning in. And if you're tuning in, on the YouTube, you missed your opportunity to ask us questions. But I'm very grateful for Shamali for joining me and for our audience here and you at home, wherever that may be. And we may see you again in another Google Hangout. So thank you for watching. Bye-bye.
Thank you. Bye-bye.